Good evening, everybody, and welcome to the Human Tissues lecture. So, no more cell, no more the cell membrane, no more the cytoplasm and the organelles and the cytosol, no more the nucleus, the DNA, the transcription, the translation, functional proteins, structural proteins. We discussed all of that. Now it's time to move up one stage upward. So what comes up from the cell? Tissues. Students tell me that I ask them to define a tissue. They say when you have different cells. Can a tissue have different types of cells? No. The organ can have different types of tissues. That makes a lot of sense. The organ system can have different types of organs. That's fine. But the tissue can have only one type of cells. And we said how many different types of tissues do we have in our bodies? Four. Nervous and muscular, they come together because they are excitable. They respond to electrical stimulation. And then you have the connective tissue, which is the most variable of all the tissues, the most widespread. It has different types of connective tissue we're going to learn about. There's the adipose tissue, fat tissue. That's connective tissue. Your blood. Somebody came up to me with a blood smear. That is a type of a connective tissue. You have the areolar tissue, the reticular tissue of your spleen. So that's, and lymph nodes. That's an example of a connective tissue. And lastly, you have our favorite, the epithelial tissue. Now that one is very diverse because it comes in different cell types and different relationships to the epithelial tissue comes in two different relationships. One is the cell type, and the other one is the relationship to the basement membrane. Okay, so when we talk about the tissues no matter, even though that they are four, epithelial tissue, connective tissue, muscular tissue, and nervous tissue, no matter what type of tissue it is, in the embryological development, it had to have come from one of three places. Either the ectoderm, meaning the outside layer, outer layer, ectoderm, or the mesoderm, that's the middle layer, the mesoderm, or the endoderm, that's the innermost layer. You can be able to tell which tissue comes from which layer. For example, your skin. Do you think your skin, who's all the way on the outside, on the external layer, do you think that comes from the ectoderm, the outside, or the middle, the mesoderm, or the innermost layer, the endoderm? They come from the ectoderm and they stay there. So you see that the nervous tissue and the epidermis arise from the ectoderm. What about the mesoderm, the middle layer? When you get a needle injection, the needle feels painful when it's at the surface of your skin because there's the nerves, so many nerves on your skin. But once it goes to the middle layer, which is muscle and bone, do you feel pain now? Do you feel pain when it goes into the muscle and the bone? You don't feel pain because the bone and the muscle tissue are the middle layer. Your skin is the outermost layer. The innermost layer, we have a hollow tube. That's why they do an endoscopy. So they put the camera in this hollow tube. So we are hollow from the outside and we are hollow from the inside. So that internal layer that has your mesenchyme, and it gives rise to the mucosa and the collagen of the inner layer is the mucous membranes, we said, the digestive and the respiratory tract. So everywhere where there's mucus, that's the endoderm. So if we want to examine our tissues, we discussed in the past that you would have never known that a particular egg, that egg, if you did not slice it in half, you will never be able to tell that there is yellow yolk on the inside. So same thing with our bodily tissues. If you did not splice and cut your humerus coronally, frontally, split it in half. Will you have ever known that inside the core of this bony tissue there is red bone marrow, blood in there? Would you have ever known that? Would you have ever known that there's yellow bone marrow, butter, lipid, cholesterol in there? So you have to do a complete cross section. And we're going to look at this tissue from different cross sections. There's a longitudinal section, there's a oblique section, and we're going to begin now with our epithelial tissue. This epithelial tissue, remember, we do both. We study the structure and function. Structure and function. So when we begin with the epithelial tissue, we're going to look at the structure of the epithelial tissue and the function of the epithelial tissue. So the epithelial tissue structure, anatomy, it's a sheet of closely adhering cells where you have one or more cells that are thick, and there is an upper surface that is exposed to the environment or the internal space. So you see that the epithelium is either on the outside or the inside. I'm sorry, what's happening in the back? Please have a seat and pay attention to the lecture. Stop the computer and pay attention to the lecture, please. Yeah, so don't stand, don't interact, just pay attention, look there, please. So this epithelial tissue is an ectoderm on the outer layer of the skin and on the inside of your mucosa. Now this different type of epithelial tissue, recognizing that this epithelial tissue is just epithelial tissue is not enough. This is where the differentiation happens. You see, on the skin, you're going to have squamous. You're going to have squamous tissue that is stratified, very thick. But in the esophagus, are you going to have stratified squamous tissue? You're going to need modification like the ciliated cells. Those ciliated cells have the cilia like a power stroke and a recovery stroke. Power stroke and a recovery stroke to propel the mucus layer away from the respiratory tract and prevent any mucus plug. And this mucus plug is very dangerous if it wasn't for this epithelial ciliated modification. So that type of tissue belongs there because it wouldn't be any other type of cell. Connective tissue, muscular tissue, and the nervous tissue do not have the ability to operate like columnar ciliated tissue that's going to remove this mucus. So patients with cystic fibrosis, they suffer a lot with this mucus plug because there is an overwhelming amount of mucus buildup that's going to be in the respiratory tract. So that's an overwhelming amount than the cilia is able to remove from the respiratory tract. So we begin structure, function. Structure comes before the function. Anatomy comes before the physiology. Not just because of the alphabet, A comes before P, but actually, if we don't know the structure, we're never going to make a connection with the function. So the structure of epithelial tissue is that it's a sheet of closely adhering, stapled cells with Gabby junctions. And you see that with the upper surface, they're exposed to the environment, or from the inside to the internal. There's an external space, and in the alimentary tract and the respiratory tract, there's an internal space also. It covers the body surfaces and most of the glands. And this epithelial tissue is avascular, meaning that itself doesn't have red blood, it doesn't have blood vessels. But does that mean the basement membrane? So how do they stay alive? If this tissue, this epithelial tissue is avascular, doesn't have blood vessels, how does it stay alive? So the basement membrane is very rich in blood vessels. So the epithelial tissue, even though it's avascular, but it is reliant on the Basement membrane, very good. So this basement membrane is the source of blood supply and vasculature to this epithelial tissue. Now we finish with the structure of the epithelial tissue. Now we look at the function. The function is to protect the tissues from injury. The epithelial tissue, we said, is responsible for making up the glands, so they produce and release the mucus, the sweat, the enzymes, the hormones, and it's important for secretion and excretion. What's the difference between secretion and excretion? So secretion is when the glands release useful substance, like enzymes, like hormones. But excretion is when we get rid of waste. Which one is more significant, secretion or excretion? They're equally, yeah. They're both important. You don't want any buildup. If anything, I would say excretion is more important. You can survive with the problem of secretion, like diabetes. You can get exogenous. You don't have an endogenous gland supplying your internal environment. You can get an exogenous medication. But excretion is the problem, is to get rid of this buildup of metabolic defect, metabolic waste. Absorption, they absorb the chemicals and the nutrients in your intestine. That is epithelial tissue. Filtration in your kidney. All the substance leaving the body is filtered by the epithelium. When we do the renal system in AMP2, you will see that the glomerulus, this filter structure, strainer, it has a lot of epithelia that is able to perform this filtration. Sensation, there are nerve endings in the epithelia that are not part of the epithelial tissue, but they are part of the basement membrane. That's why when you get a needle, you feel the pain. So they rest on the basement membrane, and this is what we discussed earlier. We examine our cell shapes and epithelial types based on two categories: the morphology and the anatomy and the structure of the cell that is an epithelial cell. We said we have three types of epithelial cell types. The first type is sunny side up, egg. That's a squamous epithelial cell. Then we have a cuboidal epithelial cell, where what equals what? Yeah, the length equals the width. And you have a columnar epithelial cell, where what is more than what? Length is more than the width. And then that's not enough. We also have to examine how are these cells aligned in relationship to the basement membrane. If you have one layer of epithelial cells, no matter if they're squamous, cuboidal, or columnar, but they're only one layer, no more than that, what do we call this type of cell tissue based on the basement membrane? Simple epithelium. What if you have a mixture where some cells are touching the basement membrane and some cells are on some cells? So if you look at stratified, the cells are either touching the basement membrane or touching each other. But in pseudo-stratified, some are touching the membrane and at the same time they are touching other cells. So you see one cell is arranged in a matter that is pseudo-stratified. Is that clear? So we have simple, pseudo-stratified, and stratified epithelium. Okay. Now we said there is simple, so now we begin with the simple epithelium. Simple epithelium, you have three types: simple squamous, simple cuboidal, and simple columnar. I talked about these wine glass shaped cells known as the goblet cell. These goblet cells are known for doing what? They are known for secreting mucus. So now these are found where? These goblet cells. They are found in the Yeah, what type of tissue? Yeah, you're right, in the esophagus, but where? Simple, columnar, and pseudostratified epithelia. That's a simple columnar modified with pseudostratified epithelia. That's a special type of cell. So now we're going to examine every and each one of them. So the first combo, what's the first combo? Simple squamous. Simple squamous. When you hear simple squamous, you don't even need to look. If you examine simple squamous epithelium, you don't even need to look at the microscope. You can already have a mental picture of what simple squamous epithelium tissue looks like. What does simple squamous mean? So that means all the epithelial tissue cells are on one layer on the basement membrane. There's many types of membranes out there. On the basement membrane, when all the epithelial cells are attached as a one layer to the basement membrane, that is simple epithelium. But it's not only simple, it's simple squamous. So what kind of cells are those? Yeah, so they're nucleus facing up. There you go. Do you see now? Do you see where my mouse is? Look, that's an example of one layer. You see, they're only one layer of cells, linear layer of cells. And they are not only one layer, they are egg sunny side up. You see the egg sunny side up. So now this slide shows you the structure. They are single row of thin cells. That's the anatomy, the structure. Now what's left? The physiology. They, number one, permit rapid diffusion, transport of substances. They secrete serous fluid. And their locations, where are they found? Where are they found? They found in the alveoli. They're found in the glomeruli. They're found in the endothelium, and they're also found in the serosa. They're found in the serosa. Okay, so let us now examine this special type of tissue. Simple cuboidal epithelium. Simple cuboidal epithelium. From the name, if you examine simple cuboidal epithelium from the name, you have simple cuboidal. That means all the epithelial tissue cells of the simple cuboidal epithelium. Simple, meaning what? That means all the cells are what? Are one layer only on the basement membrane, like a village with only one story houses, no two stories, or no two or three or four. So that's in like the rural areas. So you have one layer of simple. If it was stratified, it's like skyscraper, or very urban type of design. So you have like 3 or 4 story buildings. So now if you look at simple, that's simple. Cuboidal, meaning that the width and the length are equal. So now let's look at where we find that type of tissue. You have single layer, because it's simple, square or round cells. So that was the structure, square or round, single layer. Now they are important for the absorption, secretion, mucus production, and movement. Now, what's the location of the simple cuboidal epithelium? Number one, we find it in the liver, we find it in the thyroid, we find it in the salivary gland, the bronchioles, and the kidney tubules. Simple columnar epithelium. Simple columnar epithelium. You have a single row of tall, narrow cells. You have a single row of tall, narrow cells. So this is a simple columnar epithelium. Simple meaning you have only one layer, one row that is attached to the basement membrane. And in addition to that, they're not only simple, they are columnar, meaning that the cells all have a length that is much more long, much more than the width. You see the columns here? What's the name of these wine glass appearance types of cells? These are the goblet cells. And they are known for doing what? These goblet cells? Mucus secretion. So you see here you have a single row of tall, narrow cells. There's an oval nuclei in the basal half of the cell. There's a brush border of microvilli, sometimes ciliated, and they may possess goblet cells. And the absorption and the secretion, they are important for absorption and secretion of the mucus. And they are lining, where are they found? In the GI tract, in the uterus, the kidney, and the uterine tubes. Ciliated pseudostratified. These are, from the name, ciliated, it means that they have a modification. They have an extra type of an addition. So the ciliated pseudostratified columnar epithelium, ciliated meaning that it has cilia, and cilia is known for which strokes? The power stroke and the recovery stroke. And that is important for doing what? Mucus, moving the mucus and preventing the formation of mucus plugs. So now, if we look at the pseudostratified, some cells, you see here, they're pseudostratified. They're on top of each other, but some of them actually make it all the way to the basement membrane. So they look multi-layered, and they all touch the basement membrane. The nuclei are at several layers. They have cilia and goblet cells, and they secrete and propel the mucus, and their locations are the respiratory tract and portions of the male urethra. Stratified epithelia, they range from 2 to 20 or more layers of the cell. So now we're moving from simple into stratified. We have finished simple, and now we're dealing with stratified. There's a special type of stratified tissue known as the urothelium. Someone talked about that earlier in the lab. You see, that's one of the perks of doing lab before lecture. You get exposed to this urothelium, which is a form of a transitional epithelium. It is found in the urinary tract. You need to know that. Where do we usually find transitional epithelium? We find it in the urinary tract. So these stratified epithelia are named for their shapes. So some are stratified squamous, some are stratified cuboidal, and others are stratified columnar. Stratified columnar is a rare and is rare and only found in places where 2 or more epithelial types meet. And it can have the same way the simple columnar ciliated, it had cilia, it was simple columnar and had a modified cilia. Stratified can have modification too, but is it going to have a cilia? It's not going to have a cilia. What is it going to have? It's going to have keratin. You need to know that the simple tissue can have a modification when we add cilia, but stratified will have a modification where we add keratin. So some are keratinized, cornified. These are the desquamating and exfoliating cells of your skin. These are found on the skin surface, and they are abrasion resistant. They are non-keratinized. They are found lining the internal spaces, and they lack the surface layer of the dead cells. Keratinized stratified squamous epithelium. You see the picture shows you where is that found. So keratinized stratified squamous epithelium, that's an example of a multiple cell layer where cells become flat and scaly toward the surface, and they resist the abrasion, and they retard the water loss through the skin, and they resist the penetration by the pathogenic organisms. The location of where we find the keratinized stratified squamous epithelium is on the epidermis, in the soles of the feet and in the palm. So this is just a matter of you have to sit down. You have to sit down and go over all these variety of tissues and examine their anatomy, their structure, and their physiology. How do they work, and where we find them? Where do we find that type of tissue? Non-keratinized, that's also stratified squamous. So all the cells are egg. So if you look at the non-keratinized stratified squamous epithelium, all the cells are like an egg sunny side up, and you see that that's a keratinized epithelium without the surface layer of these dead cells. So there's no keratin. Where do we find that? In the tongue. Imagine there is keratin in your tongue. It will be very dry, not going to be able to do the job. So that's still stratified squamous, but it is non-keratinized. Very good. In the oral mucosa and the esophagus. How are we doing anticlockwise? Oh no. I don't know. I'm trying. Okay, stratified cuboidal epithelium. So we have the cubes, but they are stratified on top of each other. So cuboidal cells in the length and width equal, and they are stratified. So they are packed on top of one another. So there are two or more cell layers, and these are surface cells that are square or round. They secrete the sweat, so that's their physiology, that's their function now. They're important for secreting the sweat, production of the sperm, spermatogenesis, the making of the sperms, and producing the ovarian hormones. And their locations are sweat gland, ducts, ovarian follicles, and the seminiferous tubules. Is that clear? The urothelium is an example of multi-layered epithelium with the surface cells that change from round to flat when they are stretched. That's a special type of tissue we all need to be familiar with. The urothelium represents a multi-layered epithelium with surface cells that change from round to flat when they are stretched. They allow for the filling of the urinary tract, and their locations are the ureter and the bladder. Ureter and the bladder. So the connective tissue overview. So the connective tissue is the most abundant, widely distributed, and variable type of tissue in which the cells occupy less space than the matrix. So we're now done with the epithelial tissue. Now we're dealing with the connective tissue. So here most of the cells are not in direct contact with each other. That is the hallmark of connective tissue. It's not stapled, it's not glued to each other like the epithelial tissue. Epithelial tissue is hooked with hooks, these cell adhesion proteins, cell adhesion molecules. So they're widely distributed and they have a variable type of tissue in which the cells occupy less space than the matrix. Most cells are not in direct contact with each other, and they have a variety of vasculature. Some adipose tissue, some connective tissue is highly vascularized, and some connective tissue is not vascularized. So, for example, the loose connective tissue has been very enriched with blood vessels, but the cartilage has no blood vessel, even though both are a type of connective tissue. You see, some connective tissue is highly vascularized, like the loose connective tissue, and cartilage has no blood vessels on it. So that's very interesting. It's not like the epithelial tissue where all of it is what? All of it is avascular. Very good. All of it is avascular. But the connective tissue, some of it is vascular, some of it, because